A very beautiful morning to you all, and welcome to yet another session of Educare Analytics as powered by Safe Publications. In this session, we shall harmonize the queries on gene technology as sent in by several advanced students in our inbox. Let's kick off. Gene technology refers to the molecular techniques, procedures, or methods used to manipulate the DNA of organisms and thus their proteins to obtain the desired outcome. The desired outcome can be study purpose, like for research or utilization due to applicability of the formed protein. It is also known as nucleic acid biotechnology since it uses both DNA and RNA to manipulate the organism's genome. However, the use of RNA involves locating the RNA-based sequence, which is an exact copy of the gene, and then converted into complementary DNA, that is cDNA, using reverse transcriptase enzyme, as we shall discuss later. The manipulation of the organism's genome, that is DNA, is commonly known as genetic engineering. Therefore, gene technology was formerly or traditionally known as genetic engineering, but the former is broader than the latter as it includes modern techniques like gene editing and other fields like synthetic biology. Ideally, it involves isolating a gene of a desired trait in one organism and transfer it into another organism using a suitable vector or method where the gene is expressed. The transfer of the genes or gene can occur between organisms of the same or different species. So, gene technology eliminates restrictions or barriers of species specificity. That is, a human gene can be expressed in a plant so that the plant starts to produce or synthesize the human protein. The DNA of the transferred gene is called donor DNA, and it is inserted into the host DNA of the recipient. The resultant DNA containing the donor DNA or gene transferred and the host DNA is known as the recombinant DNA, rDNA. The organism which expresses the new genes of the rDNA is called genetically modified organism, GMOs. GMOs are also known as transgenic organisms. Terms used in gene technology. A gene is a sequence of nucleotide bases of DNA that codes for a specific protein. In other words, it's a section or segment of DNA that codes for a particular protein. Restriction enzyme, specifically restriction endonuclease, is an enzyme that cuts DNA at a specific site called restriction site to DNA fragments with sticky ends. The restriction site is a base sequence like GAA, TTC. The restriction endonuclease cuts the DNA each time a specific base sequence occurs. That is, if the sequence occurs 5 times, then the restriction endonuclease makes 5 cuts, forming 6 DNA fragments. DNA ligase is an enzyme that joins DNA fragments or sections. A vector is an element that transfers a gene from one cell to another. Examples are plasmids and viruses, phages. Plasmids is a small circular extrachromosomal bacterial DNA. Stick ends, that is staggered ends, are short length of unpaired DNA molecule formed when a restriction enzyme cuts the DNA. That is, are short length of unpaired DNA strands. They allow foreign DNA to bond more easily with non-stick ends, that is blunt ends. Recombinant DNA, that is rDNA, is the DNA made by artificially joining together pieces of DNA from 2 or more organisms of the same or different species. Transgenic organism is an organism containing foreign DNA from another source, such as another organism of the same or different species. In other words, it is an organism whose DNA was changed artificially. It is also known as genetically modified organism, GMO. However, the GMO can be obtained by selective breeding, unlike a transgenic organism. Gene probe is a length of DNA with a base sequence complementary to another piece of DNA someone intends to detect. Genome is the complete set of genes present in a cell or organism. For eukaryotes, it includes nuclear DNA and mitochondrial DNA, as well as chloroplast DNA for plants. Genetic marker or marker gene is a sequence of nucleotide bases of DNA linked to the desired gene to give evidence that the desired gene is incorporated into the host cell. That is, it is used to indicate transformed cells. Bloating is the transfer or movement of nucleic acid or protein from one medium to another. It is usually from gel to cellulose or nitrocellulose or nylon membrane. It is southern blot if you are transferring DNA, northern blot if you are moving RNA, and western blot if you are transferring a protein. There are several gene technological techniques, but our scope is limited to only 3 techniques, namely recombinant DNA technology, gene cloning, and polymerase chain reaction (PCR). Recombinant DNA technology. This is a molecular technique used to combine DNA from different sources, cell or organisms, to form new DNA known as rDNA. rDNA molecule is a chimeric molecule formed by joining foreign DNA with vector DNA, that is, DNA containing genetic material from 2 different organisms. Recombinant DNA technology involves the following steps. DNA isolation. The DNA fragment representing a desired gene is identified in the donor DNA using gene markers and isolated or extracted from the donor genome or cells using appropriate methods like centrifugation, chromatography, and precipitation. Cutting DNA. The donor DNA is cut into small DNA fragments with stick ends using restriction enzymes. Likewise, the host DNA is also cut by the same restriction enzymes to form complementary stick ends, which easily allow the donor DNA of the desired gene to be ligated or spliced into the host DNA within the host cells or the vector. Ligation. The stick ends of the donor DNA and host DNA are joined together by DNA ligases to form recombinant DNA. Cloning. The rDNA is then introduced into another cell or organism called the host cell or organism using the appropriate vector such as plasmids, phages, or other suitable methods like electroporation or microinjection, among others. Expression. The host cell or organism then expresses the inserted gene by producing the desired protein coded for by the foreign gene of interest. The produced proteins on expression have various commercial applications: enzymes, hormones, interferons, and antibiotics, among other medicinal proteins. These applications will be fully discussed later. Because of our time, let's pause it here as we wait to dissect gene cloning in our next session. For details on this segment of gene technology and more, kindly refer to our biology book with comprehensive notes for advanced level titled ACS Biology Kindle Volume 2, or visit our YouTube channel for exclusive discussions. Thank you for learning with us.